Hey guys, so let's talk about gene regulation. And once again, don't want to get into too much details on both of them, but what I want to talk about is the contrast, something that may not be clear from PowerPoint or videos, but should be clear in your mind. So let's talk about the first thing first. Prokaryotes and eukaryotes. What is the main difference between those two? And I can hear you scream, organelles. Yes, it is. It is a main difference. Prokaryotes don't have them. Eukaryotes do have them. And that's what leads to the main difference in gene regulation. Think about it for a second. In the PowerPoint, in the videos, they tell you prokaryotes, there is that like operon, the example we use in the PowerPoint. So what is a like operon? It's simply along the sequence of DNA, there is not only genes for one simple protein, but it's just a sequence of gene on that DNA strand, and on that DNA strand you start with usually, let's look at the red, you usually have a promoter, then you have an operator, and then you have like the genes. Because remember, in prokaryotes, here, the entire DNA is usually circular. At some point it closes out, but it doesn't have several chromosomes, it doesn't have several molecules of DNA. It's usually only one. And so the promoter is going to just help promoting, promoting the activity of the expression of those genes. But sometimes, and that's where the DNA polymerase starts here on the promoter. But the operator, that's where repressor proteins can come here. And those repressor proteins, we may have remember from the operant that those repressor protein prevents the giant molecule of RNA polymerase, the protein of RNA polymerase, from moving, sticking here, cannot go to the genes, cannot express the genes. And that's pretty much it. Like, you can have RNA being made. Imagine the repressor is taken away by the presence of lactose, for example. You make RNA. And so, what else do you have after that to regulate that RNA in prokaryotes? Well, you can deteriorate that RNA. You can destroy it, right? You can just digest it. We don't have lysosome, but you can digest it anyway. You can break it down with enzymes. Yeah, that's one way. When your proteins are made, you can just prevent the folding of them, you can prevent the moving of them, you can destroy them. Well, that's cool, but really you limit it in what you can do. Now, if you look at eukaryotes with their organelle system, and most importantly, the nucleus being that giant organelle in the center of the cell that has all those nuclear pores. The nuclear pores only RNA can live, right? RNA is small enough, DNA is too big. So we do the transcription, we get RNA, and RNA can live, right? Either way they want. Well, it turns out you can just prevent that. You can just clog those pores with proteins, all the stuff. You can prevent the RNA from living altogether. You could also interfere with the processing of the RNA in the nucleus. Remember that the RNA has introns and exons, and that the parts that are not coding for protein will be removed. You remember that you have to have a cap and a tail on the little RNA molecule to prevent degradation. Those can be forgotten and not added. So lots of ways already, and we haven't left the nucleus, but you can just do things differently, right? Now that your RNA has been processed, let's say everything goes well, it leaves the nucleus. Once you in the cytoplasm, now your little molecule of RNA needs to go and find ribosome. Ribosomes is either floating, as it would in the prokaryote, or it's on the endoplasmic reticulum, right? And if you remember, when you have that endoplasmic reticulum, and it's going to be the rough endoplasmic reticulum, RER, and I'm just going to represent like one side of it, that's like the inside here, and that's the outside of it here. When that pieces of RNA goes and attach itself to a ribosome on the surface of that rough ER, that RNA is read, and protein is made inside the ER. That protein here, its primary structure. The rough ER is where the secondary structure and the tertiary structure and the quaternary structure gets together when it gets packaged. Well, if in the ER you don't package it efficiently, if you don't fold it efficiently, right? All of that is another way to regulate the genes from which the RNA is coming from, right? That's RNA. So another type of regulation in eukaryotes is due to another organelle, or rough ER. It can be to the Golgi apparatus. You can have lysosome chiming in and destroying things along the way. So there are lots and lots of regulation that can be done in eukaryotes compared to prokaryotes. And the main difference, it's organelles. None? Yes.